Hi, my name is Liam. I work at the Centre for Proteomic and Genomic Research. I went to UCT Medical Campus, joined Professor Jonathan Blackburn's lab for my Masters, which I then upgraded to my PhD, and that was in medical biochemistry. And that's when I was introduced to proteomics as a technology. And it was then that we decided to use this technology to look at an interesting phenomenon involving patients who have TB and HIV and who receive their treatment but end up having quite a severe adverse reaction. It's known as TB-related immune reconstitution inflammatory syndrome. I run the proteomics platform at the CPGR. I have two other colleagues who work with me, Michelle and Shanae. And together we try and help clients understand systems level what's going on with their samples using the proteomics technologies. The predominant one in our platform is LC-MS, making use of Orbitrap technology. The best way I can try and describe what we do is, if you were a racing car driver and you made use of a Formula 1 car, you would get the most out of the performance of what that car could do. If you were a regular everyday driver, you wouldn't really be able to come close to what that car could do. And in many ways, our job is to try and take the racing car, which is our instrumentation, and drive it as if we're Formula 1 drivers. So use our expertise in how to make sure that it's running efficiently, running really nicely, quality controls are all working well, and generate good quality data that can come out of that and be used for sort of systems level analyses. That's really fundamentally what we try and achieve. And we try and do this as either sort of like a CRO type function, so an outsourced R&D type of function for big companies, or within the academic space to try and facilitate academics who may not have the expertise in this area but want to make use of this technology for their particular research focus. Big data generally is becoming more and more important. In many ways I view this as a bit of a currency, and proteomics allows you to generate large amounts of data quite quickly, and it's generated at the coalface effectively. So if you think about your biology, you have your genes, which, a large-scale study of genes would be your genome. Genes become your RNA transcripts, and a large-scale study of that would be a transcriptome. And from there you get your proteins. A large-scale study of that would be your proteome. And so the proteome really is where everything happens in the cell. They're the doers, they move things around, they build the physical structures, they're really involved in driving a lot of the mechanics and the mechanisms that occur within cells, within tissues and between organs. It's really, really important to get an understanding of what's going on there. So this can have implications in all kinds of fields, whether they're medical, agricultural, marine associated. It's really very, very vast. We often have to rely on the expertise of the researchers that we work with to bring in the biological understanding of what's going on, whereas we can help them on the technical side to make sure that what they're looking at is of high quality and really, really good.